Hi, my name is Dongjoon Lim, and I want to present our work on Graphylo, a deep learning approach for predicting transcription factor binding sites from whole genome multiple alignments. The process of transcribing DNA to mRNA is very complex. It often requires involvement of different proteins, known as the transcription factors. These transcription factors are the major player in gene regulation, and they bind to short specific patterns in the DNA called motifs that are usually 6 to 15 base pairs long. The transcription factor binding sites in the DNA are thus critical components of gene regulatory networks. There are other factors that affect the transcription factor binding to the DNA. It includes epigenetic state of the DNA region, for example, the chromatin accessibility, DNA methylation, and 3D genomic organization, etc. The presence of other transcription factors bound to the neighboring sites also results in the cooperative or competitive interaction with the transcription factor that we're interested in. What is the exact definition of transcription factor binding site prediction using computational models? The experiment used for identifying regions bound by transcription factors is chromatin immunoprecipitation followed by sequencing, also called the ChIP-seq experiment, which is a very common technique for identifying genomic locations bound by a given transcription factor in a given cell type. The ChIP-seq data can be used to train computational models that predict transcription factor binding sites in silico. And these models can provide nucleotide level resolution and insights into sequence specificity and architecture of multi-transcription factor binding regions, such as promoters and enhancers in the DNA. The computational predictors are also important for interpreting the potential consequences of sequence variants. So to formulate this problem, these models given a genomic sequence S and the transcription factor T try to predict whether T will bind to S. What are some existing computational models for transcription factor binding prediction, and what are the challenges that they're facing? The first computational model for predicting transcription factor binding to the DNA are using position weight matrices, which is a representation of motif profile by calculating the frequency of nucleotides in the regions in the DNA that are thought to be bound by transcription factors. And after the advent of the deep learning era, many deep learning approaches based on the neural networks trained on the ChIP-seq peak dataset were introduced. It includes the DeepBind that uses human sequence for predicting. FactorNet and DeepGRN uses both human sequence and the epigenetic experimental data as an input to predict the transcription factor. These kind of models still face some challenges that these epigenetic experimental data, such as DNA-seq, is not always available. So there are methods using multiple sequence alignment data, which is almost always available, for complementing missing functional information based on the motivation that the interspecies conservation is highly correlated with the function of a sequence. These models include phylogenetic footprinting and phylo-PGM. Extracting features from the multiple sequence alignment within the context of evolution is still a challenging job. So this is the exact problem we're trying to solve, with the motivation that orthologous sequences give useful information of sequence function. Our objective is to train a supervised deep learning model that predicts the transcription factor binding site, given a genomic sequence S in the human, and its orthologous sequences in 115 species, a phylogenetic tree T that represents the relationship between these species, to predict whether a transcription factor will bind to this human sequence S or not. The model input includes the multiple sequence alignment blocks of 201 base pairs and its reverse complement, plus the phylogenetic tree that represents the relationship between the species in the multiple sequence alignment. The exact pre-processing step includes isolating DNA sequences of placental mammals from a 100-way multi-Z alignment of whole vertebrate genome. We then infer maximum likelihood ancestral sequences using the Ancestor 1.0 program. The labels are obtained from the ENCODE-DREAM in vivo transcription factor binding site prediction challenge ChIP-seq data, and we are sampling the equal number of positive and negative samples. This is the architecture of the model we named Graphylo. We first leverage the power of convolutional neural networks, which enable us to capture important features in the multiple sequence alignment for both the forward strand and then the reverse complement. The features extracted for the reverse complement are then flipped left to right and are stacked together with the forward strands feature to form a very rich feature set. Then the species attention module will be attending species-wise attention to our representation for each species, and this scaled representation of each species is aggregated by the species that are located in the neighboring node in the phylogenetic tree using graph convolutional networks. The node embeddings of this graph convolutional networks are then flattened out and passes through a fully connected layer to make a prediction whether this multiple sequence alignment block is bound by a transcription factor or not. We evaluated the performance of Graphylo on the DREAM within cell type transcription factor binding site challenge and compared it against several other models that uses different types of input data. Our comparison includes two versions of FactorNet, which is a CNN stacked LSTM model that uses either human sequence alone or human sequence with the FastCons conservation score. We also compare it against RNA tracker that employs a CNN, LSTM, and attention mechanism, and the PhyloPGM, which is a probabilistic graphical model that uses predictions made on different species in the phylogenetic tree and aggregate to make a better prediction on the human DNA. We can see that 9 out of 13 cell types in this data set, Graphylo is the best performing predictor, and the p-value is lower than 0.01 for the Wilcoxon signed ranked test. So this Graphylo is significantly outperforming other competitors that use different types of input data. Here, we compare 3 different models that uses evolutionary information for making transcription factor binding site prediction, compared to the FactorNet baseline, where it only uses a human sequence for making a prediction. So we have examples in different FastCons conservation score bin. When we look at the examples where the FastCons score is the lowest, all 3 models gain only a little bit of performance boost AUPR. However, when these conservation scores gets larger, which means the example is more conserved among different species, we can see that the performance gain Graphylo is having is significantly larger than the performance gain the other models like PhyloPGM or FactorNet plus FastCon is having. So we can conclude that Graphylo successfully incorporates the phylogenetic evolutionary information better than the other models to make better predictions on the transcription factor binding sites. We then analyze the attention weights learned by the model to understand the role of different species in the prediction process. By examining the relative importance of species, we can better understand which ones have the greatest influence on the final result. We discovered that there are 3 distinct patterns of attention allocation across different transcription factor and cell type combinations. The first pattern highlights data sets where the majority of modern species and the ancestral species receive notable attention values, indicating that the predictor is utilizing information from the entire tree. The second cluster displays the transcription factor and cell type combination, where attention is primarily focused on the human sequence and then the most recent common ancestor of human and chimp sequence, but not on the other sequences. The final cluster reveals dataset where the majority of the modern sequences are given attention, while most ancestral sequences are disregarded. Well, further follow-up studies are needed to better explain these attention wastes and how the evolutionary information is affecting the transcription factor binding site. To summarize, Graphylo outperforms its competitors by a significant margin, improving the median AUPR scores by over 5% on 13 different ChIP-seq dataset. The future research includes combining the Graphylo architecture with approaches that also take input cell type specific experimental data and investigating versions of Graphylo where sequence information is only provided at the leaves. For the current version of Graphylo is quite slow, but a sped up predictor could be obtained by excluding species from the analysis with low attention values. Graphylos can also be applied to other types of sequence function predictions and may prove powerful at predicting the impact of mutations in these types of regions. Graphylo has a potential to provide a more nuanced understanding of the functional importance of different mutations. Thank you for listening to our presentation, and we thank all the members of the Blanchet Lab for providing useful advice on the presentation, and we also thank the funding agencies.